Uh, Okay, so let me start off with kind of giving an overview of what I'll show. Um, so I'm from Converge Health by Deloitte. We were part of Recombinant prior and helped do a lot of the original development for tranSMART. But one of our most recent projects was with the FDA. And until now, when doing your analyses in tranSMART, you could only do one study at a time. So if you wanted to compare patients across studies for different reasons, you really didn't have a good way to do that unless you're just doing each analysis in one study at a time and then comparing the results. So what we enabled for the FDA was the ability to start to actually look at data across trials in one analysis. And what they wanted to do was, since they were getting um, device information and drug information and vaccine information for all sorts of different areas that would potentially treat the same disease. What they wanted to be able to do was compare treatment options to find out if they were more effective than others for specific outcomes, or look at adverse events or safety issues and determine if there's drugs that actually are safer or result in fewer adverse events overall. Um, they had a lot of focus actually on devices, um, but they also were focused on, like I said, vaccines and drugs as well. So we did this project with the FDA about a year and a half ago, and just to acknowledge who was part of the team, we had a team from Converge Health, mentioning everyone on the left there. And then as for the folks at the FDA, we engaged with the Centers for Device drugs and for vaccinations, and CDRH, CBER, and CDR are those three centers. Um, I couldn't remember everybody in the list, so at risk of forgetting somebody, I just wanted to acknowledge the centers themselves. Uh, so with that, what I'll do is I'll show you a little bit about what we've done. And I'm going to start off and show you an instance of tranSMART that is not 1.2, but it was the foundation for what we've done in 1.2. And the reason for that is because I just wasn't able to load enough studies into 1.2 to really show off the features. But I will show you how it works in the new version as well. So why don't I get started here, and I can show you, this is Dataset Explorer, now called the Analyze tab. And in my particular case, I'm going to look at asthma and COPD patients. So in this case, what I might want to do is just compare asthma and COPD patients across multiple studies. And to give you an idea of what studies are loaded, I can click on the studies. You can see I have a lot of breast cancer data, but I have 2 asthma studies and 2 COPD studies that are listed here. So it's going to look across both those studies when I do summary statistics. And I get the standard histograms back for age and for the pie charts for sex. And I'll also see race information. In this particular case, no race data was collected for these particular studies, so that's why that's not there. But then if I want to drag other information in, just like you would do in a tranSMART looking at a single study at a time, I can drag in something like smoking behavior and see how many patients were ex-smokers, non-smokers, or smokers in both sets of studies. So I'm looking at 4 studies simultaneously here. Now this is all well and good that I can get some idea of this information. And one thing to note when looking at this is I can see that, for instance, my second subset here, which are my COPD patients, they're a bit older than my overall population for asthma patients. So if I want to do a more in-depth analysis on this, and I can look at, I'll do a box plot in a moment, but I want to look at one of their lab findings associated with their disease, I'm going to limit the age to folks who are just over 45 to make sure that I'm comparing a similar population. So I'll come back to the comparison tab, I'll clear what I'm doing, and now I'll just have my population be patients who are over 45 years old. And I'll move into an advanced workflow. I'll start off with box plot. That's the one I'll be demoing today. And what I want to determine now is there a difference in one of these lab findings based on the condition that the patient is in, or what the diagnosis of the patient is. So I'm going to look at the different lab tests. I can see I've got forced expiratory volume, which is a good measure of lung capacity and lung health. And I want to look at that with the various trial arms and see any difference amongst these four studies that I've got here. So I'm going to drag all these trial arms in. And I'll just run this as is. And what that'll populate back is a box plot looking at all of these different areas for my forced expiratory volume. And you can see it's a little bit all over the map, as you might expect, but you can also see that my COPD patients have the lowest volume. So I can see this across all these different studies. Now, to take this one step further, if I wanted to group these, I could actually rerun this analysis and use binning. I can manually bin the groups. I'm going to manually bin the dependent variable, I believe. Excuse me, the independent variable, which is a categorical variable. And what I want to do is I'll put all my healthy controls into one. I'll put COPDs in maybe bin 4 over here. The folks that don't have COPD are also qualified as healthy controls. And then we've got maybe one non-severe asthma, and I think the rest of these are all considered, actually, I think one severe asthma, the rest of all these are considered non-severe. So then now I'm looking across multiple studies, and I run the same analysis, and I'll just come back with just 4 plots for the box plot, and I can see that there's definitely a trend associated with the categories I just described. So that's the idea behind what we enabled for cross-study analyses and boxplot. I'll show you now how that works in version 1.2. It's very, very similar to what I just showed. The only difference is it's only going to be going across one study, or because that's all we were able to load for test purposes. So if I look at 1.2, in the navigate terms section, instead of having a cross trials on the right, it's just below here. And I could drill in just as you saw Jeek do a few minutes ago with all the different areas. Now for cross trials, I'll drill here. And in this particular case, I'll actually substitute my population by those lab tests in this case. So what I can do now is look at forced expiratory volume, do that by a numeric value. I want to say something like 80 is a good number to divide up this group somewhere in the middle there. And then I'll just run the summary statistics the same way I did before, and I come back with all the same graphics. And this would be going across all the studies loaded within the Across Trials tab. I could also move in and go into box plot and do the same thing I just did. Actually, let me clear this first. So if I clear this and just bring in all of my patients, for instance, if I just bring in, if I look at interventions, for instance, or if I look at studies, maybe in this case, I think if I just look at one study, I can also bring in a second study if that were there. I think in this case, I actually only have data loaded in the first study that's shown here. But now I can do that same box plot that I just described, where I can bring in the various trial arms, look at forced expiratory volume, and run. And it comes back with a very similar graphic to what I just showed. Again, it's only limited by the data right now. But that was all I wanted to show today. So if there's any questions on that, let me know. I know it was a much shorter demo than what we just saw, but hopefully it makes sense and everyone understands it. Any questions?